to welcome everyone to the Hearn webinar series. For those of you that may be joining us for the first time, I wanted to introduce you to Hearn. My name is John Cadiz. I'm a part of Hearn. I help run the coordinating center. You can see from the logos here that this network is an NIDDK funded network, and it was initiated back in 2014 with two major objectives in mind. First, to conduct research investigating the loss of human beta cells in type 1 diabetes, and then second, to identify strategies to protect and replace functional beta cell mass. Today's webinar is co-sponsored by the NIDDK Information Network, or Dknet, which again is another NIDDK funded initiative, and we'll get back to that in a minute. So the way NIDDK decided to tackle these major objectives was to create a modular network of different consortiums. And what you see pictured here is all of the different consortiums that have been created over the years. Each has a different theme. We recently published an editorial that gives you a little bit more context and history for the network. But in brief, it's a large international effort across several institutions, many different countries, bringing together a lot of the top T1D scientists in the world. And if you want to learn more about it, we obviously have a website and you can gain access to a number of different resources. But I just want to highlight one of those in particular, which is what we call our resource browser. And what this is a tool where we catalog all of the different bioreagents, data sets, documents, technologies that Hearn is using and or generating. And this includes different antibodies, mouse strains, constructs, data sets, protocols, lots of different resources that you can go to and actually find here on the website. So I'd invite you to go and check that out. Regarding today's webinar, if you miss any part of it, it will be posted on our Hearn YouTube channel and also on the Dknet website. So don't worry, it'll be there. You can revisit it. To tell you more about Dknet, I'd like to invite Ko-Wei to join us. Ko-Wei, are you there?
Yes. So hello, everyone. I'm Ko-Wei Lin. I'm the project manager of the Dknet. As John just introduced, Dknet stands for NIDDK Information Network. And it's our pleasure today to co-host this webinar with Hearn. And for those who are new to Dknet, it's a research resource information portal funded by NIDDK. We provide several services, including resource and tools that support rigor and reproducibility. We also help researchers discover useful research resources and keep up to date on new tools.
So essentially, they get away with it. Yeah.
Can you please mute yourself? I'm sorry, Ko-Wei, please go ahead.
Okay, and we also have the Hypothesis Center to assist researchers in finding new tutorials for public available online resources and in generating research hypotheses, and a fair data management service to help make your data fair. So in addition to these services, we have funding opportunity for new investigator and a summer scholarship opportunity for students who work in lab during summer conducting research topics in NIDDK relevant disease field. We also have webinars to introduce useful resources and tools, and a monthly newsletter. So if you're interested, please visit dknet.org or follow us on Twitter or sign up to our newsletter. So I have two short announcements before the webinar starts. Our next webinar will be on Friday, May 13. Dr. Kenneth Young will give a presentation on AI approaches to predict type 1 diabetes. And our summer program, I just mentioned, the student will have scholarship opportunity. If you have student or you are a student who work in an NIDDK relevant disease field such as diabetes, the application deadline will be on next Monday, May 2nd. So you can find more information from the website. We'll start our webinar today. I will turn the mic to John. John, over to you.
Thank you so much, Koei. I appreciate it. What you see in front of you are the topics that Dr. Joglekar will cover. I won't read those out to you, but what I will do is remind you to please check to make sure that you're muted so that there are no disruptions as we listen to Dr. Joglekar give the today's presentation. I'm excited to present him to you today. I will stop sharing my screen right now, and he will go ahead and share his screen as I introduce him. Dr. Joglekar is an assistant professor in the Department of Immunology at the University of Pittsburgh. He did his postdoc at Caltech and PhD at UCLA. Alok's work involves looking at the landscape of antigen-specific T-cell responses in health and disease, and then using that information to manipulate the immune system to fight disease. Alok joined Hearn in 2020 as a recipient of the NIDDK Gateway Investigator Award. And then later in 2021, Alok was also the recipient of an award from the Dknet's pilot program in bioinformatics. So he's been very active in these networks, and I'm excited to see what he's going to be sharing with us today. Thank you, Alok, and I look forward to your talk today.
All right. Can you hear me well, and can you see the right slides?
Yes, we can see your opening slide, and I can hear you fine.
Awesome. Thank you, John and Koei, for the wonderful introduction. Of course, thank you to NIDDK, Hearn, and Dknet for funding our research and giving me the opportunity to interact with colleagues in this field. And thank you all for joining this webinar. So today I'll talk about a really exciting area in immunology in general, but type 1 diabetes in specific, which is T-cell antigen discovery and how we can leverage T-cell antigen discovery for research in type 1 diabetes. So before we jump into the specifics of the T-cells and T-cells in type 1 diabetes, what I want to do is first give you an outline of the webinar. So we'll start with sort of defining the challenge that we are facing here, which is how do we analyze T-cell responses in different disease contexts? And then I'll go over 3 different approaches to identify T-cell epitopes. 2 of those approaches are sort of experimental approaches, and then the last approach is a computational approach, and then we'll go over sort of the pros and cons of each of those methods and how to really choose the method that's right for you. Then I'll briefly talk about how we can integrate the data on T-cell antigen specificity with other multi-omic data sets. And then finally, I'll show a couple of brief snippets of how we're using these methods to study the role of T-cells in type 1 diabetes in our lab. So let's first start by appreciating what the immune system does. So on a daily basis, our immune system is challenged with lots of different threats. These threats can come from bacteria, viruses, parasites, tumor cells, all sorts of different things. And the immune system has built several sort of responses and barriers to protect us against these threats. These include physical barriers like skin and mucosal membranes. They include large-scale non-specific type of responses, such as the complement system. And they also include precise responses that are generated by the adaptive immune system that can act in a very targeted manner. So together, through these 3 different sort of barriers to protect us against threats, the immune system deals with a large number of challenges. And our work in particular, and that's the topic of today's webinar, focuses on the adaptive immune system. And some of the defining characteristics of the adaptive immune system, as opposed to the innate immune system, are that the adaptive immune system can respond in a targeted manner, but also in an inducible manner where it can rapidly scale up a response to a particular challenge. It has immunological memory, so if the immune system is faced with the same challenge again, it can respond to it quite rapidly. There are, of course, multiple layers of regulation which both help make the adaptive immune system more effective, but also less or limits its pathogenicity. But one of the key features of adaptive immune system, which my lab is particularly interested in, is the antigen specificity of it. So the adaptive immune system has the ability to distinguish self and non-self, and of course, different types of self and non-self molecules. And this is where our research focuses on. So T cells form one of the key components of the adaptive immune system, and the key characteristics of T cells which make them either protective or pathogenic in different diseases is their antigen specificity. So T cells use their surface T cell receptor in order to recognize peptide epitopes presented on MHC molecules. So a T cell receptor consists of TCR alpha and beta chains that recognize the MHC and the epitope, and it consists of gamma, delta, epsilon, and zeta chains, which form the signaling components. And canonically, T cell receptors on CD8 positive T cells recognize epitopes presented on class 1 MHC molecules, and T cell receptors from CD4 positive T cells recognize epitopes on class 2 MHC molecules. So what are the MHC molecules actually showing to these T cells? So both class 1 and class 1 MHC molecules or complexes, and by the way, they're known as HLA molecules in humans. Both these complexes actually sample the intracellular and extracellular proteome and then present peptides from those proteomes to T cells. So class 1 complexes typically present peptides from proteins that are translated within a cell, so an intracellular sampling as such, whereas class 2 MHC complexes can present peptides from exogenously acquired antigens. So it's estimated that together, class 1 and 2 MHC complexes present anywhere from 10 to the 4 to 10 to the 6 molecules on the surface of cells, depending on the context. So what's available to T cells to see is this enormous and highly diverse universe of antigens. So how do T cells actually recognize and respond to all these different antigens? And the answer is that they generate an equally enormous diversity of T cell receptors. And the way they do that is through a process known as VDJ recombination, in which fragments of T cell receptor genes, so for example, variable and constant fragments, are joined together through a combinatorial process that leads to an enormous diversity of T cell receptors. And it's actually estimated that our immune system can contain anywhere up to, or even somewhat beyond, 10 to the 9 unique T cell receptor clones. What does it mean when I say a unique T cell receptor clone? So a T cell receptor has, as I mentioned, alpha and beta chains. And for both chains, there are variable regions which are typically involved in antigen and MHC binding. And these variable regions are formed from V alpha, J alpha fragments for the alpha chain, and V beta, D beta, and J beta fragments for the beta chain. And together, these encode 3 hypervariable regions known as complementarity determining regions, or CDRs. And typically, CDRs 1 and 2 bind the MHC, and they're actually germline encoded in the V fragments, whereas CDR3 regions actually bind kind of both epitope and the MHC, and those are generated through the process of VDJ recombination, and they're not germline encoded. So through this process of generating an enormous diversity, the T cell repertoire is then able to respond to these threats. So going beyond the repertoire, the T cells have sort of if you can think about it, a relatively linear program where a T cell will recognize its cognate epitope using its TCR. It'll initiate a signaling cascade within the T cell. That signaling cascade will lead to changes in gene regulation of certain genes, which will lead to expression of certain genes. And these genes would be then, or the gene products of these genes would be then used as a way to functionally respond towards that epitope. So over the years, there have been many methods that have been developed to look at all the different functions of a T cell. So for example, to study T cell repertoire, one can use single cell RNA-seq or bulk sequencing of V beta. We can also use RNA-seq methods or chip-seq type of methods to look at gene expression and regulation. We can use phosphoproteomics and proteomics-based methods to look at signaling and functional responses of T cells. So the question, or really the challenge, is how do we integrate the function of a T cell at both a single cell level and a repertoire level? And one of the keys to that is a rapidly advancing field, which is focused on actually sequencing the T cell receptors. So because T cell receptors are what target or what determine what the target of a T cell is, sequencing or knowing the protein sequence of the T cell receptors is key to understanding what their target is. So there are many methods that can be used to sequence a T cell receptor, and these include bulk sequencing methods such as Adaptive Biotechnologies immunoseq, which can sequence specific V beta regions, or they can include single cell sequencing methods, which include 10x genomic single cell sequencing with VDJ enrichment. Okay, so now we have TCR sequences and we have ways to study the function of T cells, but how do we get from the TCR sequence or knowing T cells to their antigen specificity? And this is where the bulk of the webinar will be focused on. So there are roughly 3 approaches that people take to identify T cell specificities. These are, the first approach is what's called an antigen-directed approach. So we can start with a set of antigens and ask whether we can identify T cells that can recognize that antigen of interest. Or there are approaches that are called TCR-directed approaches. So we can start with T cells of interest, and then can we identify what antigens those T cells are recognizing, starting with the T cells. And the third, and this is one of the most recent areas, is computational antigen discovery, where we can start with TCR sequences, and can we predict the antigen specificity purely based on the TCR sequences. And I'll go over different methods that are part of these 3 approaches. And this is not meant to be an exhaustive list of methods. So I sincerely apologize if I have not covered certain methods, or if I have not covered your favorite method of antigen discovery. Okay, so let's start with antigen-directed approaches. So these are probably the most widely used methods for looking at T cell specificities. And the most perhaps simplest method or simplest set of methods is to look at functional readouts in response to an antigen. So here the concept is that you start with a polyclonal population of T cells and stimulate those T cells with antigens of interest. These could be proteins, these could be lysates, these could be peptides. And then you look at which or how those T cells are responding to those antigens. And that can be done in multiple different ways. That can be done through looking at cell proliferation in response to an antigen. or activation of surface markers or intracellular cytokine production in response to those peptides or proteins, or quantification of secreted effector functions, such as by doing ELISAs or ELISpots in response to certain proteins. Now, these methods are perhaps most commonly used, and that's because they have certain advantages. So the advantages of these methods are they're typically highly sensitive. You can detect quite low frequencies of T cells. They're relatively easy to implement. There are lots of commercial kits available for that. You don't really need to know the HLA type of the patient or really much about the patient or the organism from which the T cells are derived. These methods are typically rapid, and they can also be quite quantitative or at least semi-quantitative. However, their disadvantages are that because you're looking at stimulation, you can only use a small number of peptides or proteins at once, and other than getting a response or not, there's really no other information that you can glean from this. So the other set of antigen-directed methods address some of these challenges, and these are what's known as peptide MHC multimers. And I'm sure a lot of you have heard of peptide MHC tetramers. And what these are basically soluble peptide MHC complexes that are presenting a defined peptide and are conjugated to some sort of a tag. And that tag allows us to detect T cells that are binding to that peptide MHC complex. And then these tags could be fluorophores to be used for flow cytometry or sorting. They could be heavy metal tags to be used for cytoph. They could be DNA barcodes to be used for sequencing-based methods. Or they could be something like magnetic nanoparticles, which you can use to isolate or magnetically select cells that bind to a particular peptide MHC complex. Now, these methods, as I mentioned, can help in quantifying cells directly that are recognizing a certain epitope, but they can also be used to isolate those cells. And you can also multiplex other immune phenotyping methods, such as looking at surface markers, along with these tetramers or multimers. However, the disadvantages often are that the scale of peptides that you can test at once is still restricted. Even the best methods are getting somewhere in the range of hundreds of peptides at best. They're also quite expensive to make and also non-trivial to make. They typically present only high affinity peptides, and they're not particularly great for looking at class 2 MHC complexes. So to address these antigen-directed challenges, people have developed TCR-based methods. And here, again, we start with TCR sequences or T cells of interest, and then ask, can we identify their antigens? So the first set of TCR-directed methods that I'll talk about are what's called truly degenerate or combinatorial methods. So these methods don't assume anything about the reactivity of a T cell other than a putative MHC, and use combinatorial or degenerate libraries, such as combinatorial peptide libraries, in which other than one position, all the other positions in a given epitope are completely made degenerate. And it's a mixture of amino acid. So it's called a sequential positional enrichment, where you can look at specific amino acids at each position in an epitope. Or these could be large-scale libraries such as yeast display or baculoviral display libraries, where enormous peptide MHC complex libraries that are truly degenerate are generated and then used to screen TCRs. And typically what ends up happening is that you get hits from both these type of methods, but those hits are not the actual epitope or physiological epitopes. So one would need to do some sort of a sequence homology search to look at what's the biological epitope that's targeted by those T cells. Of course, these methods, because they're truly degenerate, they allow a really high scale of epitopes, so up to 10 to the 8 epitopes, and you don't need any a priori information about the antigen. All you need is a TCR sequence to start with. However, they're quite labor intensive and cost intensive. And as I said, they often don't yield the exact epitopes, but rather yield what are called mimotopes, which then you would need to look for the biological equivalence of. They often rely, particularly yeast display libraries, rely on soluble TCR production, which is highly sort of non-robust. And because in these, the peptide MHC complexes are actually displayed on the surface of yeast, they're non-physiological because they're not in their setting of being presented on the cell. And typically, these methods are less robust for class 2 MHC restricted antigens. So to address some of these issues, particularly in the last 3 to 4 years, several techniques have come out that use cell-based methods for TCR-directed antigen discovery. The principle here is that you start with a library of some sort of reporter cells that have genetically encoded epitopes, and then you screen that library against a TCR or T cells under investigation. And then if you can mark cells that are recognized by a given T cell or a given T cell receptors, then you can isolate those cells. And then because the epitopes are genetically linked, you simply have to sequence the epitopes, and then you'll know what's the cognate epitope. And there are several methods that have come out sort of in a spike within the last 3 to 4 years. These include engineered receptors, including MCR TCRs and Sabers, which were actually developed by my lab. They also use methods in which peptide MHC complexes are actually used to target T cells for viral entry. These came out pretty much in the last couple of weeks, but also methods that transfer some sort of a tag from T cells to the antigen presenting cells. So a method that's based on trogocytosis, which actually my lab participated in, and also a method named T-scan, which transfers granzyme molecules between T cells and antigen presenting cells. So through this, one can use cell-based libraries in order to identify epitopes that are recognized by T cells. And the advantage here is the peptide MHC presentation is physiological because it's on the surface of a cell. It also offers flexible scale, so one can go from hundreds to hundreds of thousands of epitope. And because you can custom design the libraries that you're presenting on these cells, you can actually identify the actual biological epitopes of the TCRs. And these methods are often flexible to be used for both class 1 and class 2. And really all you need to start with are TCR sequences. The disadvantages are that these methods are still quite labor and cost intensive. And you typically need some a priori information about what the potential sphere of antigens could be. And then there may be affinity issues where they may be biased towards higher affinity epitopes. All right. So the last set of approaches are focused on computational prediction of T cell specificities. So here, can we start with a TCR sequence and purely based on TCR sequence or any other sort of computational or bioinformatics data, can we predict the antigen specificity of T cells? In the first set of methods, they were really geared towards being able to predict specific being able to predict specific epitopes and MHC restriction of T cell receptors. So the paradigm here is that TCRs that share antigen specificity or recognize the same antigens often have similar sequences or similar CDR3 sequences. So a corollary of that would be that TCRs that have similar CDR3 sequences may share antigen specificity. And based on that, particularly two methods that I want to highlight are glyph and TCR-dist, which were developed, in which and more methods such as cluster and iSmart and Gianna were also developed along similar lines, where different features of the TCR sequences, so for example, motif enrichment within TCRs, or TCR distances between highly similar or variable TCRs, or some sort of a feature-based clustering of TCRs is used to predict specificity. However, the caveat of these methods is that in absence of any known antigen-specific TCRs in the dataset, they cannot predict antigen specificity de novo. But where they can be helpful is that if you identify there is a cluster of TCRs that's highly similar, and one of those TCRs is a known antigen-specific TCR, then you can hypothesize reasonably that remaining TCRs in that cluster are recognizing that the same antigen. On the other hand, one can take sort of a step back and look at whether anything in TCR repertoires can bias it towards antigen specificity or towards a disease stage. So this is where TCR repertoire features can be used to distinguish between different disease states. And typically these methods start with a training data set in which there are antigen or disease specific T cell repertoires and non-specific repertoires that are analyzed together in a training data set. And these data sets are used to then these data sets are used as inputs for machine learning, where various machine learning methods are used to learn what are specific sort of classifiers based on features of the TCRs. And these could be many different methods such as neural networks, variational autoencoders, memory networks. Many different machine learning methods are particularly are being used in the last couple of years, in which one can learn what is special about certain antigen or disease-specific T cell repertoires, and based on that, can we classify disease versus healthy samples, for example, or antigen-specific versus non-specific cells. However, even though they cannot directly predict epitope specificity, they can actually distinguish disease states even without knowing what that antigen specificity is. So what I've shown you so far are three different approaches to look at or identify specificities or T cell antigen recognition. So how do you choose what antigen discovery method is right for you? And right off the top, I'm going to say that computational antigen discovery, although it's advancing very, very rapidly, it's not at a stage where one can immediately start using it for identifying epitopes. I have no doubts that it's going to get there within the next couple of years. But for this particular slide, what I'm going to focus on is between the experimental approaches, which approaches might be right for you based on what you are interested in. So this is sort of a summary of the different methods that we went over. And you can find this in our Nature Methods review that was published in 2020. And then, as you can see, the different methods shown here differ in their scale versus specificity or sensitivity of detection. They differ in their speed and ease of implementation. And they also differ in their cost or how wide they are in terms of how many MHCs or how many TCRs can be used at the same time. So let's start with, what do you know about the potential antigen for the project that you have in mind? So if you have absolutely no idea what a given TCR or TCR of interest is recognizing, or T cells of interest are recognizing, or maybe recognizing, such as alloreactive TCRs, for example, you can start with, or you can look at yeast display or combinatorial peptide libraries, because they're truly degenerate. If you have some idea of what the TCRs might be recognizing, so for example, TCRs that are known to react to certain tissues, you can use cell-based methods. But if you have a narrow range of antigens, so for example, if you want to look at pathogen-specific TCRs, then functional screens or multimers are the right methods. It also changes based on where are the T cells coming from. So if you only have TCR sequences or a small number of T cells, you can start with the cell-based or TCR-directed methods. But if you have good accessibility or availability of tissues, then you can probably use some of the antigen-directed methods. And based on how many T cells you're looking at, if you're looking at very small number of T cells, for example, TCRs that are clonally expanded or are important in a given disease, then probably yeast display type of methods are good. If you have, again, cell-based methods are also good in that sort of a setting. But if you are looking at a large number of T cells, like polyclonal T cell populations, but a narrow range of antigens, you can use the antigen-directed methods. And lastly, what I want to quickly show is some of the recent approaches that one can use to integrate antigen specificity with transcriptome or multi-omic data sets. And I'm going to just highlight two approaches. One of them is for use with sort of TCR-directed methods, and the other one is an antigen-directed approach. So the first approach I'm going to highlight is recently came out from Paul Thomas and Phil Bradley's labs, and it's called Conga, in which one can take single-cell RNA-seq data, which includes gene expression and TCR sequences, and cluster cells based on both gene expression and features in the TCR sequences. And then these clusters can be sort of correlated against each other in order to get clusters that are functionally important based on their transcriptome, but also might share antigen specificity. And then if you have the TCR sequence information, you can use those for TCR-directed antigen discovery methods to integrate the specificity into your analysis. On the other hand, a recent method called TET-TCR-seq also came out, in which you can generate relatively large, I'm talking about tens to hundreds of peptides, where you can make barcoded DNA multimers, and then you can stain T cells directly with those barcoded multimers, and then perform 10x genomic sequencing. So here you're not really getting TCR sequence, but you're getting direct antigen specificity based on the tetramers, and you're getting gene expression data, which you can integrate together. So to summarize this first part, we've now defined that T-cell antigen discovery is a fast-growing field, particularly given how many TCR sequences are being generated and deposited. And of course, there's a need to incorporate those specificities with other multi-omic measurements. We went over two different experimental approaches to identify T-cell epitopes. And a very rough guide is that if you have a narrow range of potential antigens and large number of T cells, antigen-directed methods are better. If you have a wide range of potential antigens, but a small number of T cells, then TCR-directed methods are more appropriate. And then computational approaches, they're nascent as of now, but they're really getting more and more sophisticated with the eye on the prize that we should be able to predict TCR specificities purely based on TCR sequences. And then methods to integrate those with multi-omics are again nascent, but they can be augmented with experimental approaches. Okay, so in the last few minutes, What I'll do is showcase some of our work that's funded by both the NIDDK New Investigator Gateway Award and also the Dknet New Investigator Pilot Program in Bioinformatics. So once again, thank you to these two agencies for funding this research. So our work focuses on looking at T-cell responses in type 1 diabetes. As you all know, in type 1 diabetes, both in humans and in mouse models, infiltration of pancreatic islets by T cells, both CD4s and CD8 T cells, is central to pathogenesis of type 1 diabetes. You can actually find CD4 and CD8 positive T cells directly infiltrating the islets. So in my lab, we're using non-obese diabetic or NOD mice as a model for type 1 diabetes. NOD mice are a quite robust model of autoimmune diabetes, where by about anywhere between 12 to 16 weeks, mice spontaneously develop autoimmune diabetes, and it's mediated by leukocytes infiltrating the islets. And over time, you can see here that there is an increasing infiltration of T cells in the pancreatic islets of NOD mice. So of course, my lab's interest is in looking at what are the autoantigens that are recognized and are immunogenic in type 1 diabetes. And here, one of the advantages of NOD mice is that many of the autoantigens that have been described so far, and here I'm focusing largely on CD4 positive T cells, the autoantigens are actually shared between NOD mice and humans. So several autoantigens have been known in which we can use NOD mice as a mouse model to study antigen-specific T cells that may be diabetogenic or protective. However, despite these sort of decades of research, a significant fraction of islet-infiltrating T cells, we don't actually know what they are recognizing. We don't know the specificities. And this is where our Hearn and Dknet funded projects come in. So in my lab, we're taking TCR-directed antigen discovery approaches to identify autoantigens in NOD mice. And we start with single-cell RNA sequencing of islet-infiltrating T cells in NOD mice, and then use antigen discovery screens. So if we can identify novel antigens in mice, we can take them back to humans and see if they're actual autoantigens. Okay, so to start with, we did single-cell profiling of islet-infiltrating T cells, and due to time constraints, I won't go into details of how that was done. But essentially what we have is CD4 positive T cells that were isolated from islets of NOD mice, of 6, 8, or 10-week-old NOD mice. And these are pre-diabetic NOD mice. And for each cell, we have the transcriptome and also the TCR sequence. So we can look at what are the different phenotypes that we see. And we see a mixture of naive-like, activated, and sort of effector and also exhausted T cells in here. And based on the TCR sequence, we can actually look at what's the clonal expansion of these cells. And we see that in these activated and mixed sort of effector populations, that's where we see the highest clonal expansion. And again, this is sort of expected because we would expect that the highly expanded clones are what are responsive to antigens. So we can actually take, I won't go into the pseudo time, but we can actually take the top expanded TCR clones for each week, and you can see that they fall in these more activated clusters. So what we did in my lab is we took these top sequences. So we actually took 20 TCRs each from 8 and 10-week-old NOD mice. And these were the top expanded TCRs from those mice, and expressed them in cells to perform T-cell or TCR-directed antigen discovery. And for our TCR-based antigen discovery, we're using receptors called Sabers, which stand for signaling and antigen-presenting bifunctional receptors, which are basically MHC complexes that have a signaling domain attached to them. And what this allows us to do is that if a given peptide is presented by a Saber and it's recognized by a given T cell, there is a signal that goes into the APC. So now if we have a library of APCs presenting all sorts of different epitopes, through this signaling, we can mark which cells are recognized by a given T cell receptor or not. First, of course, we showed that Sabers work. So here we're looking at GFP expression as a result of this signaling. And then we can see that when we have a combination of the right combination of a TCR along with its epitopes. So here are two different epitopes that are recognized by a TCR called BDC 2.5, whereas a negative control TCR, 4-8 insulin, cannot recognize those epitopes. And we see that only when we have the right combination of the TCR and epitope, we get a detectable signal. So we can read successful TCR peptide MHC interactions. So based on that, we've built a 4,000 epitope Saber library in which all the epitopes are actually derived from islets. And I won't go into the details of those, but we can screen these library this library against TCRs that are of interest, and then identify the cognate epitopes. And then this is the library that was based on a study coming out of the UNN-UA lab. I won't go into the details because of time, but this library has 4,000 epitopes that are all known to be IAG7 restricted, IAG7 being the class 2 MHC allele of NOD mice. Okay, so first we can validate the library. And to do that, we use the insulin-specific 4-8 insulin TCR. And this is the epitope that's recognized by the insulin TCR. And after screening this TCR against our library, what we should expect is that any cognate epitopes that are identified should be enriched in this sort of top left corner. And as you can see through the screen, we're identifying the cognate epitope of the TCR as well as several analogs of it. So we can validate that this library actually can be used for antigen discovery. So far in our current ongoing efforts, we're applying this library to identify antigen specificities of these top 40 TCRs. And what we have so far are several TCRs that we have already de novo identified their specificities. And these are either specific to hybrid insulin peptides shown here or insulin. And then, of course, we are doing more and more antigen discovery and of course, validation studies to really define the antigenic landscape of CD4 T cells. But one of the things we can do is, because we started with TCR sequences, we can actually take, upon identifying the antigen specificity, we can take those specific TCR sequences and look at what's the gene expression signatures of the T cells that are specific to those specific antigens. And again, this is ongoing, but we are looking at latent factor-based modeling to look at gene signatures that influence or that are influenced by the antigen specificity of the T cells. So to summarize this part, we have a novel cell-based epitope discovery method that we have validated and are using for identifying the epitopes of islet-specific CD4 positive T cells in NOD mice. We are also developing this for human antigen discovery. Simultaneously, through the Dknet project, we are taking features of the TCR or T cell repertoires and looking at what are the signatures of clonally expanded or antigen-specific T cells. And of course, the ultimate goal of this would be to translate this to type 1 diabetes patients, where we can actually take either PBMCs or islet samples from patients or donors, and to predict disease states predict T cell function in those, in the hopes of developing sort of antigen-specific or antigen-targeted therapies and preventative measures. With that, I would like to conclude my talk and acknowledge the people who have done most of this work. So of course, I would like to thank my lab members, particularly Paul, who is a graduate student in the lab. He's spearheaded both the experimental and computational projects here with the help of 2 undergraduate students. Again, I would like to thank our collaborators, both at Pitt and outside Pitt, particularly members of the Hearn CMI consortium, the Hearn new investigators and Dknet new investigators, and of course, NIDDK for funding both these studies. And as a last shameless plug, my lab is interested in hiring postdocs to work on these sort of projects. So if you are interested, or if you know someone who's interested, please contact me. And with that, I would like to thank all of you for the attention, and I am open to any questions.
Thank you so much, Alok. I really appreciate that. You gave a good overview in the beginning. You took a really complicated topic and you simplified it. So thank you for helping us to ride along with you. Thank you, Kowe, for joining us as co-host. We want to open this up for questions, discussions. If you do have a question, you can raise your hand or you can unmute yourself and ask the question directly to Alok. So, just check that mute button and unmute yourself if you have any questions you'd like to ask. You can also put something in the chat as well.
This is Matthias. Can you hear me?
Yeah, hi, Matthias.
Yeah.
Hi there. I have a question that's not specific to the talk, but strategically maybe important. So we know from probably the NOD, but certainly in humans, once the beta cells are gone,
right,
this interest of the infiltrate goes away. So there has to be something antigenically from the beta cells that drives this. But what has always befuddled me, and I don't know how we would distinguish amongst these different cells, in the NOD mouse we are pretty clear that insulin drives the process after George Eisenbarth's studies. But there's, for example, these IGRP T cells. But in that case, Tom Kay did a study. While they correlate with disease, and are even activated, they contribute very little to the pathogenesis. So how are we going to resolve this type of conundrum? I just wanted to ask this because the talk was very interesting. I think it's very elegant. It goes more deeper, if I understand everything correctly in terms of the profiling. But there could be a cell that drives the process and a cell that's also activated but doesn't drive the process. And I don't think we know the difference, but maybe somebody on the call does better than me. That would be super enlightening for me.
Yeah, that's a great question. Thanks very much. And you sort of hit the nail right on the head that this is maybe the question about what T cells are doing in type 1 diabetes and which T cells are doing critical or non-critical or disease accelerating functions. And it's almost impossible to, as you're aware, it's almost impossible to study in humans, but with NOD mice, we maybe can answer that after getting specific TCRs that we know the antigen specificity. So for example, some of the BDC 2.5 work, but yeah, this is the, I would say, the question in front of the T cell in type 1 diabetes field. And actually, I'll go one step beyond, and it's not just type 1 diabetes, but several other autoimmune diseases will have the same issue when you're looking at T cells and the role of T cells in them.
Yeah, I fully agree. I think, though, with the techniques like what you showed, I think we're getting closer. I don't want to leave this was not like a hopelessness question. I think technology is getting better, and I think we can tackle this in mice as well as in humans. But I was just curious. There's another interesting thing which I don't understand. In the good old LCMV mouse, the driver cells did not increase relative to the periphery in the pancreatic islets. That has always confused me. It was very curious. And sometimes, though, in humans, we see that certain specificities seem to accumulate, like these pre-proinsulin cells, CD8s, and maybe some others. And I don't understand the rules of the game, whether cells accumulate if things get chronic, and if it's too acute, they don't have time. I don't understand that either. But there's just some general philosophical questions. I don't want to noise up things, so I'm going to shut up now. It was very interesting talk. Thank you for that.
Thank you. Appreciate it.
Thank you very much, Matthias and Alok. Jewel?
Oh, yes.
Yeah, do you have a question?
Yes, great talk. I had a quick question. Can you comment, I guess it's a two-part question. Can you comment a little bit more about how you designed your Saber constructs? I guess the follow-up question to that is, are you afraid that you might identify a pool of epitopes that might be specific to that Saber construct? Or do you envision a pool of immunodominant peptides or epitopes that might be, I guess, patient in specific?
Yeah, that's a great question. Both great questions. So the answer to the second question is sort of part of the first question, which is by designing the Saber constructs in a specific way, we can sort of get around to this this issue of either having private or public sort of epitopes. So the way we design Saber libraries is we basically start with a list of epitopes, and we encode those epitopes into oligonucleotides and then clone them into our Saber constructs. And that list of epitopes, actually, that's where the most sort of design aspects of this come in. And based on the question that you have in mind, you can custom design epitopes. So to give you an example, if we are looking at tumor neoepitope specific T cells, then we know that epitopes that are generated by non-synonymous mutations that may be patient-specific would be what we would use to design Saber libraries. But in case of a more tissue-specific study, such as the type 1 diabetes work, we would look at epitopes that are maybe known or predicted to be presented on beta cells.
I see, great. Thank you for your answer.
Thank you, Joe. Any other questions for Alok? Okay, I'm seeing none. If you've got a question, just scrolling through, please feel free to ask. While we wait, I just want to again, Alok, thank you so much for giving us a really nice overview of developing methods in the field, where we're headed, some of the challenges, and sharing some of your work with us. I wanted to let everybody know as well, we have our next Hearn webinar coming up on May 18th. Our speaker will be Decio Iziric, and many of you know him. So look for announcements on our website, and we'll be circulating some emails to remind you as well. So again, thank you everyone for your time. Thank you for your attention, and we'll see you soon. Thank you, Kowei, as well, for co-hosting this webinar with us.
Thank you, John.
Okay, all right.
Thanks, John and Kowei.